The topic for the present video is a read across approach for nitrosamine and NDSRI impurities. So as we know that there are two types of impurities for nitrosamines. Those are small nitrosamines and the higher molecular weight, or the nitrosamine impurities which have structural similarity to the API. So nitrosamine and NDSRI impurities are there, which are the core of concern. These are potent impurities and these have carcinogenic and mutagenic potential. The present video is for read across approach for determining the acceptable limits for nitrosamine and NDSRIs. So read across approach uses lines of reasoning based on the structure activity relationship, that is SAR, and read across, so that the limits can be set, and these limits are to be transparent, interpretable and acceptable limits for new nitrosamines for which no toxicological data exists. So if there is no toxicological data, then read across approach can be utilized for deriving the acceptable intake limits. Main benefits or positive points for the read across approach are like it helps for the data gap filling and ensures comprehensive safety evaluations. Then efficiency. It saves time and resources by using the existing data. Coming to the key concept for a read across approach, the key concept is that the structure activity relationship, that is SAR, and it assumes that the similar structures have similar biological activities. So as similar structures have similar biological activities, it will also have the same or similar toxicological activities. So it uses surrogate compounds, uses structurally similar compounds with known toxicity to estimate the risk. For example, your molecule is duloxetine and you are also working onto the fluoxetine. So these two APIs are similar, so their nitroso impurities might be similar. So this is just for example I am giving you that the nitroso impurities of duloxetine and nitroso impurity of fluoxetine may have similar biological activities. So these are the surrogate compounds, or these are the similar compounds, so that their activities are considered to be similar. The SAR or structure activity relationship based read across approach has a regulatory acceptance, and it is recognized by FDA and EMA for setting the acceptable intake limits for nitrosamine and NDSRI limits. Some of the other regulatory bodies and regulatory authorities are also considering and have considered the read across approach for the new nitrosamine impurities and for the impurities for which the toxical data is not existing. So it works on the basis of these surrogate compounds. Now see the overview of read across approach. So purpose is to predict the toxicological property of a chemical using data from structurally similar compounds. So it is based on the SAR, and its application is assessing the genotoxicity and carcinogenicity of nitrosamine with the limited data, and it relies on structural similarity and known mechanism of actions. So these form the basis for read across approach. Criteria for selection of the analogs. So what criteria will be there for selecting the analogs or similar compounds. So first is a structural similarity, second is mechanistic similarity, and third is data source. So structural similarity means look for similar structures, that is N nitroso group. Mechanistic similarity means evaluate the potential for DNA reactive species. Data sources are use the SAR models, use the QSAR models and literature databases for deriving the structural similarity of analogs and also for the AI limits. Now coming to the methodology for a read across approach. Step 1 is to identify the target compound. Then step 2, selection of suitable analogs. Step 3, data gathering like involving the AIMS test, enhanced AIM test, carcinogenicity study data, and then data evaluation. So data evaluation is the fourth step. Data evaluation involves quality and relevance. Step 5 is data extrapolation to predict acceptable intake limits or the AI limits. So extrapolation will be done to derive the AI limit. Now what is the regulatory framework for a read across approach? ICH M7R2 guideline is there. It encourages read across and QSAR for mutagenicity assessment. EMA and FDA guidance supports science-based evaluation of nitrosamine impurities. TTC concept or TTC-based AI limit is used alongside read across for safe exposure limit. So this is the regulatory framework. Now, what are the advantages? So as the SAR technique is used and as the read across approach uses the SAR activity based AI limits, it is time efficient. It reduces need for time consuming studies. Data utilization, it uses existing data to fill the gaps, and it is having the regulatory acceptance as the read across approach is recognized by major regulatory bodies. Now, what are the challenges? So, challenges are the data quality, structural uncertainty, and mechanistic relevance. So, dependence is there on the data and dependent on availability and reliability of the existing data. So, if the available data is not reliable or not much valuable or may not be available, that time the data quality becomes a challenge. Small structural differences may impact the toxicity. So as the SAR technique is used in the read across approach, and in this the similar structural analogs are considered, and if there is a small structural difference, it may have some different toxicity or it may not have toxicity at all. Sometimes this may happen because of the structural differences and structural uncertainty. Mechanistic relevance must ensure similar toxicological mechanisms. So, if mechanistic relevance is not there, then it will have a great impact. Then coming to the conclusion. So, in summary, read across approach is a valuable approach for assessing nitrosamine risk, especially when data is not available or data is limited. Its importance, it balances the need for thorough safety assessment with resource efficiency. Future outlook. So continued evaluation with regulatory acceptance and scientific advancement is the future outlook for the read across approach for nitrosamine impurities and NDSRI impurities. So this is regarding the read across approach for nitrosamine and NDSRI impurities. Many of the professionals, when reading the guidelines, may come across this read across approach, and That's why I found this topic as a valuable topic for the present video, and I hope this uh, video will help you to understand more about the nitrosamine impurities. In the today's pharmaceutical industry, nitrosamine impurities is a very difficult type of uh, topic to handle and very important, as the nitrosamine and NDSRI impurities are very critically evaluated by the regulatory authorities. Also, the nitrosamine related questions are always being asked in the interviews. So this video will help you to understand about the read across approach. So you can answer many questions regarding the nitrosamine impurities once you go through these videos. So I insist you to go through the videos repeatedly and watch more and more times, so that your basic concept will get clear. Thank you for watching the video.